All right, what we're going to do is work out a genetics problem that is a dihybrid cross. Okay, so in this particular case, what we're going to do is look at two traits at the same time. And in doing this, we'll pick the traits here. The two traits are going to have to do with plant height and color of the seed. Okay, so we're going to have both green seeds. Sorry, let me start this over again. Green seeds and yellow. And then for height, we're just going to have tall and short. Okay, so these are our phenotypes. Okay, phenotypes for these. So two different aspects that we're going to look at. Now, how we would start this problem off is that we would have parents of the plants that were true breeding. Okay, so we have true breeding parents. And this example will start off like this, and we'll just describe the phenotypes and with an unknown genotype. So right now we're saying we don't know which one's dominant or recessive. So let's just say we have this. Let's just say we have a plant that is has green seeds and is tall. And it's true breeding, which means if it's mated with other green tall, their offspring are green tall. And if it's mated with green tall, their offspring are green tall. And if those are mated with green tall constantly, that's the same. The other, we're going to have yellow seeded short plants, and the same thing there. If the yellow short is mated with the yellow short, their offspring are yellow short, and so on and so on and so on. So they're true breeding, but at this point we don't know if the green tall is dominant to the yellow short, so we cross them. And by crossing them, in this case, this is just our example. In this case, in the example, we end up finding out that all the offspring, 100% of them, are green tall. So now that's going to tell us something here. So we're going to say, okay, green is going to be, in this case, the dominant trait, and yellow is going to be the recessive. So now we can assign these symbols to them with a capital G for green and the little g for yellow, representing that it's recessive. The tall looks like it's going to be dominant to the short. Okay, so we have tall, the capital T, and short is going to have the lowercase t. So that means then for the genotypes, the green tall, true breeding, right, well, maybe I'll put this a different color just so we kind of emphasize it here. See what you're looking at. The green, tall, with the yellow short would have to be like this. Yellow and then short is the lowercase like that. Okay, so dominant, it's dihybrid, so there's two dominant alleles for seed color, and they're both green, and that's because it's true breeding. There are two alleles for height. In this case, they're both tall, because again, it's true breeding for tall. And the same thing over here for the other one. Yellow, two alleles. We see the yellow offspring, and they're short, also dihybrid, because they keep mating again and again and again. If we were to do this in an experiment and always find that they're the same, so that would be the same as well. So these are all homozygous. Now, in terms of the offspring here, the F1 generation, its genotype is going to be different. This plant here can give the dominant allele for green, and only that one. This one can give the recessive allele for yellow, and that's it. Now, this plant over here can give the dominant allele for tall. It only has that option. That's all it has is tall. And this one can give the short allele. So that means that while the genotype of the F1 generation is 100% green tall, the phenotype is 100% green tall. Their genotype is heterozygous. So they have the dominant and the recessive for each of them. So when doing a dihybrid cross and kind of looking at some other probabilities, what we're going to do is focus on this F1 generation now and say, let's look at some possibilities or probabilities. So let's say, for example, we asked a question, what would be the likelihood that we would ever see an individual that was if we mated these with each other, that was yellow and short again, like that parent, you know? Or what's the possibility of seeing a green short or a yellow tall? That type, the other combinations. How likely is it that we'll see these other possibilities? So what we're going to do is another test cross. Okay, we're going to take two individuals from the F1 generation and mate them with each other. They're both heterozygous. Now we're going to draw the Punnett square for it, and it's a little bit different how we're going to break this Punnett square down. In this Punnett square, what we're going to have is, so each parent genotype is going to be the same, so we'll start off with the top here. This particular parent can give one of these two alleles for color, right? So it could give green, like that. Or it could give the lowercase for yellow. Now, the thing is, it can also give the alleles for height, okay, tall or short. And then the question is going to be, so do I put the tall, you know, with the dominants together and the recessives together? That's what sometimes people think, but it's not true. They're independent of one another. So we're actually going to have two options. We're going to have this plant put into some of its alleles, the green one and the tall one, as you might expect. But we're going to see a second possibility. We're going to see the green one and the short one go into some of the gametes, like this. And then we're going to see the yellow with the tall, and then the other possibility of the yellow with the short. Okay, so we're going to see 4 possibilities, and that's supposed to be a lowercase t, like that. Hopefully you can see that. Okay, so the plant has the green allele and the yellow allele. It has both alleles for height. It has 2 alleles for height. Keep testing yourself. That's color. The green is for color and the yellow is for color. The T is for the height, tall and short. So the other parent is going to have the same genotype, so it can have the same options, right? So it can have the dominants together. It could have the dominant for color with the recessive for height. It could have the recessive for color with the dominant for height, and then it can have both recessives together. And now when we start to build this Punnett square, you're going to see it's a lot more squares, right, than the one where we did a monohybrid cross. So we start to put these together. So then we have the breeding. So that means this individual, let's say these are the males here, and this is the female. So when fertilization occurs, we're just going to start to put these together and see what all the genotypes are, and then we'll see what all the phenotypes are. So that means we put these two together, and that is green with tall. So we put those together. Fertilization occurs here. We have green with tall, short. Well, it still looks tall though. The phenotype is, but here we have we have a homozygous for color, but heterozygous for height. Here we have heterozygous now for color, but homozygous for height. So now but they're still green tall. And then last one over here, we get the green yellow, but it's still going to look green, and we get the tall short, but it's still going to look tall. So we're going to see all of these be green and tall in this first row here, 100%. Even though they're all different genotypes, they're all the same phenotype. They all appear the same. Now we'll start to see some of the new possibilities here. So here we go. We go again. Green with green still looks green. Tall with short still looks tall. So that's going to be the same. Green with green, but now short with short. So this one here is different. It's going to be green short. So that's going to be a new combination, a new phenotype that we haven't seen yet. Something different. Green with yellow still looks green. Tall with short still looks tall. That's the same as all these others. Green with yellow still looks green. Again, short with short. So now we see this. So now we see two of these green short individuals. Keep going. Green, yellow, tall, still green tall like the rest of these guys. Green, yellow, tall, short, same as the rest of these, the phenotype. Now we start to see something else, something new. We're going to see yellow, a yellow individual who's tall. That's color a little easier here, just to indicate this is different. Okay, so now it's a yellow tall. And again, we're going to have yellow tall, different genotype than this one. This one's heterozygous for height. And this one's homozygous for height, but they both appear green and tall. Last row. Green, yellow, tall, short. That's the same again as all these. They're all going to be green, tall. Here we go. Green, yellow, short, short. So now it's like these up here, right? We have their green and short, so we have a third of those. Now we're going to have yellow, yellow, tall, short. So now that's the same as these two. It's yellow, tall, right? And then this last box here, it's going to be both recessives, so it's yellow, and both recessives for height, so it is going to be short. So now this one is completely different than all the rest. It's going to be a yellow, short, like one of the original parents. So if we were to count these up, we're going to have 4, 5, 6, 7, 8, 9 who are green, tall. We're going to have 3, 1, 2, 3, using the pink here, that are green, short. We have 3 more here that are going to be yellow, short. And then there's one who's going to be, yellow tall, the last one's yellow short. These 3 are, that's yellow, and then the T is the dominant, that's tall. And then the last one, we just have one who is yellow, short. So typically for the F2 generation, generation with a dihybrid cross, we end up seeing this particular ratio for the phenotypic ratio, a 9:3 to 3:1, right? And this is how it would work out. And then as you get problems about these, you would typically go back to the Punnett square, and that's how you would solve some of the problems, or figure out the ratios or proportions of the genotypes and or the phenotypes. All right. Hopefully that helps. You should look at some of your practice problems and work through them. Once you kind of get the term set for dominant and recessive, they just simply follow the same sort of patterns. But for a dihybrid, you just have to expand it a little bit and see that they're independent of one another. So you have to have both the dominant with the recessive, the dominants together, and the recessives together in order to see all the possibilities.